online. My name is Lulu Fregoso. I'm one of the Education Outreach Assistants at the Wisconsin Energy Institute here at UW-Madison. Welcome to those of you who are returning forum attendees, and if this is your first time joining us for one of our events, we're glad that you're here. The Wisconsin Energy Institute's mission is to provide leadership on campus for multidisciplinary research, education, and outreach efforts that accelerate our transition to clean energy systems and solutions. The Forward in Energy Forum is a monthly series that brings together experts both on and off campus for cross-disciplinary dialogue, exploring important technical, social, political, and economic dimensions of a wide variety of clean energy innovations and topics. You're in luck because today's forum is going to be extra unique and fun. We'll be learning about 4 different bioenergy bugs as we witness 4 scientists engage in some friendly competition. You as the audience will be involved too. You'll have the opportunity to ask questions and vote to decide which microbe makes it to the next round. Pay close attention because whoever you choose will reign supreme as the bioenergy champion. We want to acknowledge that the land that WEI occupies, as well as all of UW-Madison, is the ancestral home of the Ho-Chunk people, who have called this land Dejok since time immemorial. We recognize and respect the inherent sovereignty of the Ho-Chunk Nation and the other 11 Native nations within the boundaries of what we now call Wisconsin. Please take time to learn more about and support the efforts of tribal nations in Wisconsin and beyond, who are leading the transition toward clean and just energy systems. Next, we would like for you to save the date for our next Forward in Energy Forum, UW-Madison Renewable Electricity Goals, What are the Options, on April 22nd, which we're co-hosting with the Office of Sustainability. For today's forum, we're going to be using Menti, which will allow all of you to vote for your favorite microbe. Before we get started, we're going to do a practice round. You can feel free to pull out your phone or your laptop. If you're in the room with us, you can enter the code up on the screen into menti.com, or you can scan the QR code. For people on Zoom, the code will be dropped in the chat. Then you can answer the question that we have up on the board. I'll give you guys a few moments to do that. Wow. Oh. Can you see it like in the iPhone? Yes. Oh yeah, you can. So 11 people. Well. To moderate today's panel, I'm pleased to introduce Kevin Myers. Kevin is a bioinformatics scientist and the lead of the computational biology group at the Wisconsin Energy Institute and the Great Lakes Bioenergy Research Center at the University of Wisconsin-Madison. As a computational and bioinformatics scientist, he and his team work with researchers to analyze large datasets, write scripts and pipelines to aid in reproducible analysis, and assist with any bioinformatic or computational biology need. We are very grateful to have him with us to set the stage and serve as referee for today's bracket. Thank you.
Thank you. Thank you, everybody, online, in person. So what we're going to do today, there's this tournament you may have heard of that starts Thursday, but that one's lame. Yeah. Right? This one is going to be fun. We're going to pit microbes against each other. But before we get to that, I wanted to give a little background and to what we're doing and where these microbes came from. So as soon as Scott, there we go. So we're part of the Great Lakes Bioenergy Research Center, the 4 scientists up here and myself. And what we do is basically this. And so basically we go from energy crops that are grown on bioenergy lands, we break them down, we make them into a slurry, we feed those to microbes, and we get microbes to make bioproducts, especially biofuels. So this is housed at UW-Madison. Our main partner institution is Michigan State University in East Lansing, and we have a number of other partnerships around the country. And we are currently in our 18th year of existence. And so all of this is super great. We take crops, break them down, feed them to microbes. But what the best part is, and I'm a little biased because I'm a microbiologist, is this last part where we use microbes. And that's what we're going to talk about today. We have a a plethora of microbes that we use in the center to try to produce these bioproducts and especially biofuels. And, you know, I've always wondered who would win in a competition. I'm sure everyone has wondered, right? You all have your favorite microbes. Mine's E. coli, it's not represented here, so I'm impartial. But I'm sure you've wondered if microbes were to fight, who would win? And we tried to get them to play basketball, but, you know, they have no, their range is really low, right? So instead, we're gonna pit them against each other in their ability to produce these bioproducts and biofuels. And so first thing, I'm gonna introduce the wonderful scientists that are gonna represent, coach the teams. Get it, March Madness, all right? So I gave an overview. So what we're gonna do is, every one of them, they're representing an individual microbe. They will give a short spiel about why their microbe is the king of all microbes, or queen, I guess microbes don't have gender. And so, which is the champion microbe, and why they love working with it. And then we're gonna open up to you guys. We want people online, people in the room, to really just, any question that you have, there's no question too small, too silly, too goofy. We wanna know what you wanna know, because you're gonna have to rank these guys. Not the scientists, they're all winners, but the microbes. So you're going to use that quiz app that we used before, and you're gonna rank and vote for the semifinals. So before, we did all the previous rounds. We're down to the final four. So you're gonna vote against these two versus these two, and then after that, we're gonna crown a champion microbe from the Great Lakes Bioenergy Research Center. So, who are these microbes? Well, these are wonderful. So we'll start, I'm gonna introduce everybody and then they'll go through individually. So first, representing coaching Novus Fungobium aromaticivorians is Dr. Avery Vilbert. Representing yeast, specifically Saccharomyces cerevisiae, is Molly Davies. Morgan Davies, sorry, you are gonna mess up my name. Zymomonas mobilis is being coached by Bailey Marshall. Representing Streptomyces, not even one species, that's how great these are, is Karen Walden. All right, so what's gonna happen now is they're going to each give a short introduction to their microbe, why they think it's awesome, and then we'll open up to you guys for more feedback. So Avery, take it away.
All right, hello everyone. I am the coach for the Great Novo Knights. And I'm gonna be talking about how our team has worked really, really hard to make Novo Knights the best bioenergy microbe. So first of all, Novo, or Novus Fungonium aromaticivorians, which is its scientific name and also an absolute mouthful. So we usually name it just Novo. Was discovered in an oil and chemical spill site. And so just like us, it needs food in order to survive and produce fuel for itself. And so what Novo has done is that instead of using the normal things like sugar and all that good stuff, which it can use, it can also take all of these sort of garbage waste compounds and use that for fuel. And what our team has also found out is that Novo has such a broad diet that it can take the waste material and streams from plant biomass that Kevin was talking about. In particular, the portion of that biomass, a little bit of woody, which we call lignin. And it can take that lignin and turn it into a bunch of different products that can be used to make water bottles, t-shirts, and even glue. And so, as you can see, the Novo Knights are unstoppable, and we're here to eat our way through waste and sludge for a greener future.
Next up is Morgan.
Right, well, I am representing Saccharomyces cerevisiae, which unfortunately does not have a full nickname, it's just a cerevisiae, but you see it everywhere. So it's tasty and it's fun, it's in our bread, it's in our beer and our wine, and it's kind of just everywhere. We've been using it for forever. But it's also considered a model organism. So we use it quite a bit in science because it's a simple eukaryote. So it's very easy to kind of understand and get to know. It's also very, very well known. We know a ton about its genome and its genes. And some would say it's even the best characterized genome. It also grows so fast. So it loves sugar and it will just kind of take off with it. And because we know it so well, and because it's so fast at growing, it's also a very easy thing to use CRISPR and to edit. So that makes it really great for biofuel. While it doesn't really like eating some of the stuff we get in biofuel, because we can edit the genome pretty easily, it will, we can add in genes in there from different organisms and from things that we've been able to determine through this thing called adaptive lab evolution. We can make it so that it can do what we want, which is very, very cool. It's also very industrially prevalent, or it's used all over the place already. So we get a lot of ethanol from it already and we can get like up to a 90% theoretical yield from that. Because it stays cool under stress. It can handle being in a low pH, can handle high percentage of alcohol, and it can handle being in different oxygen environments. And yeah, so fast, easy manipulation and evolution of our Saccharomyces cerevisiae makes it the best from turning plant products into biofuel.
Morgan is being brave and representing the sole eukaryote in this, you know, I do love bacteria. Morgan did a great job. All right, Bailey, you're up.
Great. So yes, I'm Bailey. Thank you for having me. I'm proud to be coaching Zymomonas, a happy Zymozoomer, and I've written a poem for it. I love them so much. So I'll start by reading that. Zymomonas is so green, converting waste to something clean. Corn husks to ethanol, glucose to whatever at all. Zymomonas reigns supreme. I now will convince you, of course. So first of all, Zymomonas is a phenomenal fermenter, which is captured in the video, if it works. But Zymomonas is doing a similar metabolic route that yeast does that we're all familiar with in our drinks, converting sugar to ethanol and CO2. So what you would see in the video is me pipetting right up to the very, very top, and it bubbles and it doesn't go over. But you can see that they're very happy bacteria, and they're very, familiar and active and efficient at making ethanol. Depending on the strain that you're talking about and the paper you're reading, sometimes they do a better job than yeast. Just saying. This is in their ability to make really high levels of ethanol and to withstand it in their membrane. So usually ethanol will kill things. We use it for that. But Zymomonas has cleverly evolved to produce just as much as it can tolerate, which is really high levels. Another thing about it is that it doesn't need oxygen, but it can handle it. This is a really nice quality in an industrial microbe, because if you think about growing an enormous vat of a microbe and you need to give it oxygen to get it to behave optimally, then you have to spend a lot of energy to aerate the enormous vat. With Zymomonas, it doesn't need the oxygen, it doesn't want the oxygen. So we can not use that energy for aeration and just grow the Zymomonas. How convenient. And finally, they have a very small genome. So the E. coli genome is around 4.5 million base pairs, maybe 5 million base pairs, whereas Zymomonas' genome is like 2 million base pairs. This is sort of indicative of how metabolically streamlined it is. It has no bells and whistles, cannot do fancy things, but it's like a nice starting place for us as engineers to put in the modalities that we want to do. So we're starting with a bit of a blank slate. It's doing sugar to ethanol, and then engineers like myself and other people in the room are working on adding new capabilities into Zymomonas. So I'll leave you with one more thing about it, which is like the shared history between humans and Zymomonas, which is that, you know, it makes ethanol, it makes CO2, we're making beverages with it. So it's something that people have cultivated for who knows how long to drink very safely. So like the strain that we're working with in my lab and across the GLBRC from Central America, it came off of an agave plant, and it's part of the population that was used to make a drink called pulque. I know when I grow a big vat of Zymomonas, I think it smells like pumpkin and it looks delicious. So I hope you all.
Is that what it's there?
Oh, yeah. Yeah, maybe he's drinking some. So thank you. I hope you'll.
But certainly not least, Karen.
So, hi, everyone, I'm Karen, I'm representing Zymomonas, or Streptomyces.
So taken, my family.
Yeah, take it. Well, I was, so much so. So Streptomyces was first found and really started being studied because it makes antibiotics. It's a soil microbe, and they found that when they put it with other bacteria, it would kill them, and it turned out that's because it was making antibiotics. Now, it's one of the microbes that a lot of people don't realize how much they know about. So it's in the soil. And if you've ever gone outside after it rains, you know that smell. That's Streptomyces in the soil, and they're making something called geosmin. Geosmin is an isoprenoid, and I'm going to talk about that later. So Streptomyces is scrappy. It grows in the soil. It can eat all sorts of plant material, anything that you can find in there. And that's why it's really great at being used for biofuel and bioproduct making. Because it can grow everything that you'll see in there. You don't even have to clean it up very much. It will eat leftovers whatever you give it. And it also, they have it as associated with insects. So in the soil, a lot of Streptomyces use insects kind of as their way to get around. That geosmin that you're smelling is actually a signal for insects to come and get it and take it around to new energy sources. So it's kind of like it holding out its thumb, hey, insects, we need a ride. So I'm using Streptomyces specifically to make chemicals like geosmin. There are other things that are made kind of the same way, but we use them in other pharmaceuticals, in makeup, in cleaning materials, rubber for tires. All sorts of these chemicals are made using that thing that Streptomyces already knows how to do. You just have to modify it a little bit to make this or that, and then you can do anything you want. That's Streptomyces.
So instead of a commercial break, now we're gonna open it up to everybody else. So we heard 4 great pitches for each of these microbes. But, you know, you guys are the deciding factors. So we wanted to open it up to questions online, type them into the chat, and we'll relay them in person. Just shout and the old owl camera will find you and zoom in on your beautiful face. And so I will go ahead and, are there any questions to get us started? Is anyone gonna be real brave and start off with questions? Yeah, all right.
For it. Most recent with strep, what antibiotics does it make, and is there any risk for like resistance to others? Like other organisms around it?
Becoming resistant to those antibiotics. So streptomycin is obviously one that they commonly make. And some microbes can become resistant to it, but you can also see kind of the opposite thing, where some bacillus will make a compound that actually increases the activity of the antibiotic to help Streptomyces kill things so that they can feast on what is left. So you get a really complex relationship in the soil between things that are trying to survive and things that are just kind of swooping in and being like, I'll help a little bit, take some.
So I want to expand that if I could to everyone else. So Karen, you mentioned how strep can play well with others. How do your other microbes, how do they interact well in a group, right? Microbes are rarely found all on their own unless they're in a test tube upstairs. So do you have any other comments about how well your microbes interact with other microbes?
I think Salmonus is, it does not play nice. It makes ethanol to lethal levels as quickly as possible. I think that's part of its like evolutionary strategy to really win the niche, which is these really rich sugar sats that it evolved to sort of dominate. So, yeah, that's what Zymo's up to.
How about yeast?
Yeah, I would say it probably is a similar thing, because yeah, it makes a lot of ethanol. We
Mhm.
kind of want it on its own.
So yeast and zymo are selfish. Strep is a sharing tool. Okay, all right. Novo?
Well, there's not too much known about Novo since it was just discovered in 1995, I think. So we're still learning a lot about its ability to interact with different communities and different microbial communities. So far, from what we've seen, it doesn't produce anything that could be toxic to anything else. So I'm gonna guess it plays nice, but I don't know. I haven't put it underneath that stress yet.
All right, thank you.
I have a question for all the panelists, all the lovely panelists. Thank you so much for the introductions. So how much can you manipulate the metabolism, either through genetics or through just growing it in different conditions? Can you like explain a little bit about, you know, what you usually do? How much can you manipulate its metabolism, either how much it's using stuff or what it's making? Kind of open question.
So in Novo, I can speak for Novo. I don't think it's the easiest to genetically manipulate. Like I said, we're still learning a lot about the genome and what's available. But that being said, I have been able to build its own metabolism within it. So it's pretty genetically manipulated. But I will say something like probably yeast. They can use other things like CRISPR and other genome editing tools might be a little bit easier to use than something like Novo.
Yeah, I'll piggyback off of that. Yeah, Saccharomyces cerevisiae is very easily manipulated genetically. So actually a big part of what we do is to try to get it not to make ethanol, but to make other products that are more, I guess, energy efficient. So you can throw in a bunch of different genes from other organisms or that we found through various tests, and you can get it to kind of do whatever you want.
Yeah, for Zymo, it's really making ethanol as its primary activity. So to make it do anything different, you have to engineer it. And the tools to do that are being developed sort of right now. Thankfully for me that they are coming up. The Peters Lab, who are perhaps online, has really spearheaded that. And so nowadays CRISPR is possible in Zymomonas, but 5 years ago, not so much.
And Strep has all the traditional genetic tools and CRISPR and all these other things, but it also has a really cool feature called genome shuffling, where if you have a Streptomyces that's doing one thing you want and another Streptomyces doing a different thing you want, you can chunk up the genome for the one and just transfer it large chunks of DNA at a time until you get ones that are doing that function. So you can manipulate them without knowing exactly what pieces of DNA do what you want. You see a phenotype, you can just transfer it over.
I think you had your hand up earlier.
Yeah, so, I mean, I'm an engineer, so I was kind of gonna go like, tell me about the conversion, right, of each of these guys. But that's a kind of technical question, boring. So I'll ask you a better question. Who would be the best roommate? Bread, guys may clean up, like, oil, make bread, make alcohol, or eat anything, I guess.
And the question a little louder for our online audience is,
Yeah, so
who
we
would
are-
be the best roommate?
Roommate award. I want to
Roommate
say
award.
Best roommate award. right? Like novozymes, like you clean up all greases and stuff. And
The natural cleaner here,
Right.
yeah, no, but
And the yeast makes bread and all the cookies.
here.
Zymo makes the alcohol. And then I guess it's strep.
And strep cleans up all the waste and then leaves you with a fresh rain smell.
I will say that yeast, compared to other things, I think it smells pretty good. If you like the smell of beer, I mean, maybe your apartment will kind of smell a little bit like a bar, but it's fine. And I also, a lot of people do have yeast as a roommate. Like if anyone has like a sourdough starter in their fridge, there you go. So I think it's pretty good. You can make bread whenever you want. Maybe make some moonshine, who knows?
Yeah, good answer.
I'm gonna take a few offline.
Okay,
Yeah, so first question that came is, which ones show promise for the greatest throughput, being able to scale up to large volumes?
yeah, so maybe to ask that in a slightly different way, but everybody, how would the microbes do when you, right, you can't grow everything in a test tube, when you scale it up? What do you know about your microbes' ability to be scaled up to these huge liter fermenters that we've seen? Let's start with Karen this time.
Okay, Streptomyces has already been used industrially in these big bioreactors to make antibiotics and other compounds, so it has been proven to scale up.
Can't argue with that. I believe Salmonella has never been scaled up, but it's still something that we're trying to, you know, it shows great promise. There's like good leads about why it would be a compelling organism, but you know, the history with yeast or E. coli, which have been scaled up from Streptomyces, I don't think anybody's taken the risk on Zymo yet.
Yeah, I mean, historically, Saccharomyces can be pretty well done in a large quantity. If you've ever done like a brewery tour, you can see those like giant vats. And it's pretty similar to what you were talking about in your earlier pitch about how Cerevisiae can grow in like a low oxygen environment in those big containers. So it's pretty low energy to keep them going. And it can handle all the ethanol it's making without killing it, so.
So Novo has not been used in industrial processes yet, but I will say we have some great engineers working on the Novo Knights team who are right now looking towards scaling up these reactions and getting the environments just right so that we can get full conversion from our waste to the product that we want. So that's in the process, in the work.
You wanted another one
Sure.
online real quick?
Yeah, maybe, I know, Kevin, you had that diagram before of the, you know, the different steps in the process. Maybe you could each explain a little bit like where in the process your microbe fits into that bigger picture a little bit more.
I'm gonna pick Bailey to go first.
Thank you.
Yes.
So it's this step from sugars into bioproducts and specialty biofuels. So Salmonella, like I said, it's phenomenal at making ethanol. And right now, of course, ethanol is a very useful compound for us. But we're interested in engineering it to make other things. So other alcohols like isobutanol is ongoing. Love that. My personal project is in making other bioproducts. So not alcohols, but other high value compounds, which could be pharmaceuticals, could be cosmetics. So that's the engineering that we have going on with Salmonella, that last step.
Morgan?
I think we're about the same place. We're over there. It's kind of hard to see at this angle. So, yeast needs some sort of processing to be done to the plant matter that we're getting, which is usually done by our EFL lab, very cool. But once that's broken down, we are working on getting the yeast, although it doesn't really want to make a lot of the sugars from there, because there's some glucose, but there's also some xylose, which, my boss actually says it's kind of like trying to get your kid to eat like sugar, which is glucose, or broccoli, which is xylose. So they really don't want to make the broccoli, but we're really like pushing. So once it breaks down, then it can take all the monomers and the sugars and stuff and turn it into our ethanol and our isobutanol. Through genetic manipulation.
Avery?
All right, so Novo is actually on this top portion up here, taking these lignin monomers. And so that's that woody material that I was talking about previously, which is really lignin's a tangled hot mess of what these compounds that we call aromatics. And aromatics, as you guys know, have a really nice scent to them. That's why we call them aromatics. It's what makes our nice perfume. And we take those, or Novo takes those lignin monomers and we convert it into a number of different products that can be used in production, again, clothing and water bottles, using gloves.
Karen?
So streptomyces can fit in either using lignin or sugars, but because it is so scrappy, what's not shown on here is that every step in this process, there's material left over. So after they make the lignin polymers and the polysaccharides, the stuff that they wash out that they don't use, streptomyces gets that. And after they make these lignin monomers and sugars, all the stuff left over, streptomyces gets that. And then after they make the bioproducts and the specialty biofuels, all the stuff left over after that, streptomyces gets. So I get to use all of these things to make my products, which is pretty nice. Although to be fair, there's only so much left over after the primaries get it.
All right. Want to do one in the room real quick?
Sure, why don't you-
Sam?
So a lot of you guys talk about the qualitative qualities of all of your organisms, but I want the quantitative side of it. For the more older model organisms, could you explain like what industries you've impacted severely and possibly even the number of dollars you've influenced, and for the newer model organisms, the industries that you would impact?
So to repeat the question closer to the mic is, tell us the industries that your organism has already impacted, and if you know something about the monetary value of that, and if it hasn't impacted industries, which ones could it?
Thank you.
Let's start with Morgan,
Um,
as the model, as the oldest model organism. Not you, but you're
Yeah.
5,000 year old.
5,000
my niece would say otherwise.
That was a tough game.
Yeah, yeast is being used quite regularly already industrially. I mean, for our fun foodie things and beer and whatnot, but for biofuels, it's already being used industrial. I definitely cannot give a number. But I do have a number that you can get it to a 90% of its theoretical yield of ethanol in industry. So it's pretty efficient, and I read something and I don't want to misquote, but each addition, like each manipulation we make that makes the yeast better will get a higher and higher profit for the industry. So the number is great.
Let's go to another one, that Karen, you said strep is.
Probably the next oldest model organism, yeah, 50s, so, that, we got here. So I already talked about the medicines, the antibiotics, and the things like that, which have impacted human health. A lot of the replacement chemicals that they're doing with streptomyces are meant to replace things that we normally get from plants. So a lot of the value of that is based on what the current market rate of those things are, and since they're crops, that changes from year to year.
Bailey, Rimo,
Yeah, so in terms of making ethanol, that's like sort of a technology that's well developed. Simonis, sure, we could implement it there, but it's not broke, don't fix it. On the bioproduct side, I know that Simonis could be used to make some of the things that are currently being made unsustainably. So we have like polymers coming from petroleum that Simonis could generate the monomers that make up those polymers. So I don't know the market values or the money. But from like a sustainability standpoint, Simonis has a role to play there.
and Avery Novo.
And I will piggyback off of that, what, Bailey was saying was, so what Novo is really working on is converting some of those lignin compounds to different products. We are working on things like nylon, if anyone knows nylon, as replacements. And that right now is currently, again, produced from fossil fuel sources, but it's also produced on the million metric ton scale annually. So it's a lot of these fossil fuels being sort of used in the production of this chemical. Not only that, there's a lot of really nasty byproducts that are produced as well during the synthesis. So looking towards more sustainable production of these. Again, I don't know a number off the top of my head, but that's really sort of where Novo's looking more towards, is those replacement compounds.
Great. You want to do another one?
Sure. There's a couple of questions on other things that these microbes could consume. There's a question whether they can consume CO2 or maybe algae or other biomass. Anyone have any thoughts or?
Yeah, something that came up for me today is that Simonis can fix its own nitrogen. So it doesn't need that supplemented in its media necessarily.
Can you explain what you mean by fixing nitrogen?
That's a great question. So nitrogen is in life as we know it. It's also very present in our atmosphere as inert nitrogen. And so that process of taking it from the atmospheric version into a version that is biologically active or like relevant is really energy intensive, and a lot of organisms on this planet don't do it and they just consume it. Which is to say they consume nitrogen that they can incorporate into themselves. Simonis can pull it from the air and then put it in its body.
Anyone else? The things your microbes can eat? Sounds like strep eats anything, so.
I mean, a lot of times it's isolated by literally putting paper or pieces of wood into a tube and waiting for it to eat it. Scrappy.
What about Novo? How widespread is Novo's appetite?
Novo's appetite is very broad. And we're learning new things about Novo every day. Every day I come into the lab and I'm like, oh yeah, you eat that too. That's great. And because it was isolated from this chemical spill site, essentially, it had to develop all these different ways in order to metabolize these sort of waste compounds. And so we're learning, or I'm learning every day something new that Novo's kind of chewing on, which is great.
And Saccharomyces just sticks to sugar?
You like sugar. Not getting it to eat like those other sugar, sugar compounds, but it's kind of picky. Like a kid.
Any other questions? Yeah.
Out of pure personal curiosity for strep, what insects, wood boring insects is it associated with?
So the Curry lab, which was a lab that used to be here on campus, went all around North, Central, South America, trying to isolate different streptomyces. And they found it with mud dauber wasps. They found it in cobwebs, even associated with some spiders. They found it with moths, I think, and the wood dauber as well. So several, and a honeybee. I think they found a honeybee associated one. So.
Is it good for the insects?
The insect, it doesn't hurt the insect. So streptomyces have this dormant state where they form spores. And it's kind of like you might think of a seed. So like when a bird eats a seed, it disperses it. The same with an insect eating one of these streptomyces spores to help disperse it. Safely passes through.
So I'm curious how fast your microbes grow. So let's start with Avery this time. How fast does Novo double? Maybe and the conditions that would be relevant for bioenergy, for biofuels, like.
For conditions, it's probably about 4 hours, which is pretty good.
4 hour doubling time? How about yeast?
Yeah, if it's normal glucose, it's like 3 hours, I would say, for doubling time. Xylose is a little bit longer, but
And mine depends on what it's eating too.
Yeah, so that's something.
Perfect conditions, though.
perfect conditions,
Yeah.
yeah. 3 hours, you can get a transformation done in a day.
All right.
Zymo's about 80 minutes.
Strep.
And streptomyces is probably the slowest. I grow it on waste material where a lot of the carbon's already depleted. And so it's going to take usually like 4 days to fully grow. 4 to 7, depending on what you do with that. If I get the good stuff, it can grow in a couple of days. But it's process.
Fully grow as in like reaching your
Maximum
target
OD.
OD. Okay, so that's a little different.
Then doubling time.
Okay.
So what's like the funnest fact about your microbe? Like it can just be something random, something people might not know. I don't know. Sorry.
We've already shared so many fun facts.
Zymomonas is so similar to yeast, and of course, yeast is the classic bread material. I read a paper where they said they made bread with Zymomonas.
No way.
But they didn't include any pictures. I think that's very fun.
The Pixar didn't happen, right?
Exactly.
Is there a recipe in that paper?
I think they have a protocol.
Yeah. I would say just, the coolest thing about Novo is just that it's pretty new to the scene. And so I think that that's kind of interesting. It's just where it was isolated, now what we can do with it is beautiful.
Streptomyces, I think one of the coolest things about it is how you can get it to make so many different things. You can take genes from other microbes, you can take genes from plants to try to put into it. And so it has a lot of flexibility with the things I've tried to get it to do. And I guess I expected it to be more like, no, I won't do it, I won't do it. And it's like, yeah. Your chromosome, what's your favorite part? Okay. Okay, she likes the fact that it has a linear chromosome, but many other organisms have circular chromosomes. And so it puts genes that like make these sorts of chemicals on the ends of its genome, so it can randomly duplicate and make lots of copies, so then it makes lots of products. So it can control how much of the products it makes kind of on its own.
That is a very fun fact.
Thanks.
Anything about yeast
Coach Bailey there.
with the assist?
Yeah.
Basketball term.
I feel like there's so much known already about yeast that I think the best part about it is just how just like tasty it is. Really hard not to drink the stuff coming out of the lab.
All right. Any online questions?
Yeah, we got it. I
I
mean,
said nuts. I'm not drinking.
I'll combine a couple here. So one question is, what limitations do each microbe run into and ways to overcome them? And then if the microbes had an arch enemy, what would it be and why?
Limitations, I'll call on Karen first.
Oh, limitations. So limitations, it can make a whole bunch of different things. So it sometimes likes to make a whole bunch of different things because it's in the soil where it might have to respond to invading microbes or insects or water conditions or how much oxygen or all these other things. It has a really complex internal system of trying to figure out what it should be doing and when it should be doing it. And sometimes in the lab, that means it wants to do what it wants to do randomly. So you have to really work with it to get it focused on what you want it to be doing.
Avery.
Yeah, I would say that one of the limitations with Novo is just how little we know about its genome and what it can metabolize. So one of the sort of things that I'll run up against is if I'm trying to make one product and I'll sort of force it to be going towards that one product. Novo will just decide now that it can take any of those little compounds in between and funnel that or eat it and start consuming it. And so that's something that for me has been a little bit of a limitation. Again, on the learning side, we're learning more of what this microorganism can consume. But on this other side, it can pop up during when I'm trying to make 100% yield of a product.
I mean, I would say if yeast has a nemesis, it's probably me. Like it wants its sugar and it wants to make its ethanol and just go for it. But we really, that's also kind of one of the limitations is that it's got a one-track mind and we're really trying to push it to do other things, but you'll hit some roadblocks for you. It'll just be like, nope, I'm done. I don't want to do it anymore. I've got to find a different way. So I don't know, constructive criticism, I guess.
Yeah, I think Zymo's limitation is its limited diet. Yeah, when we try to feed it Zymos, it's like giving a kid broccoli, very challenging to make it eat it. Yeah.
Morgan answered the second question, arch enemy. I want everyone else to answer that too, because that's a great question. So I'm gonna have Karen go again first. Who's the arch enemy of streptomyces?
I would have to say that it would be fungi because it grows kind of the same way in soil where it reaches out and tries to break apart plant tissues. And it also can eat those really complex, difficult things. So they're usually going after the same food sources in the same ways.
Bailey?
I'll say its nemesis is variety. It just wants sugar.
And Avery?
I mean, I kind of like Morgan's response. I do feel like I am forcing Novo sometimes to do things and Novo is also just not having it. So, you know.
Be stuck in its ways.
It can, yes, be a little stubborn.
This is curious. So if you have sugars and you put all of these together, after, I guess that Zymo idea will make a quantified task, right? But after it's over, will you start attacking like or eat or like the rest of those stuff? Like,
Yeah, maybe that's, I'll combine that with a question online is, kind of what happens when you want to harvest the thing? What happens to the microbe? Can you still use it again or what does it do?
So Novo will eat the sugar that we give it and those aromatic, those nice smelling lignin compounds at the same time. So how we design it is that all of those compounds, those lignin aromatic compounds are going towards the product that we want. So it still needs some other carbon source of food to be able to eat and grow. But after it's done producing the product that we want, then we kill it. You can't reuse it over again. Yep.
Morgan?
Kind of a hard question to answer because I've only ever done it in kind of a lab experiment where you do just unfortunately send it to the autoclave.
Trey just joined and said you could feed the yeast at the end of the fermentation to cows, so that's
Oh, that's something I did not know. But if you keep giving it sugar, it will keep growing for quite a while, so. I guess if you passage it should be fun for a good amount of time.
Yeah, so if you give Zymo sort of like the complex media that comes out of the plant hydrolysate, it would eat what it wants and it would leave the rest. So maybe another organism would like to eat it. But then the Zymo would be disposed of.
Strep.
So when we get the waste material after the different steps, there is usually dead yeast or dead zymo in it that has, well, I don't tell her what to do.
That
So.
looks so mean.
The only one that's.
I know, good try. So it will do that. It does like to just keep eating the things, but yeah, eventually it's gonna run out of food and then go into that dormant spore state.
So I interpreted this question as if they were all in the same vat or media, which one would win? So like would Zymo produce too much ethanol and kill the rest, or would Novo like have this, that, and the other?
Or strep sporulate.
Yeah.
Sporulate, just hang out.
Cockroach.
I'm a little afraid of strep. I don't know if
Yeast buffers really well.
So we have to test it.
Yeah.
Novo cannot grow in greater than like 2% of ethanol, so I'm pretty sure it would be gone. I would hate it.
If Zymo and yeast can like make ethanol before strep like really grows, I think we'd be okay, but.
Until the end.
Pick a strep that's making antibiotics.
Yeah.
I'm not sure about that one.
Any more online? Any more in here? I think we're about ready to vote, right? So it might be perfect timing. Before we vote, I want to thank all the panelists. We're not done yet, right? We have to actually do the competition because we have a crown.
Oh,
For the winner.
Beautiful.
So first, you're gonna sign on to this Mentimeter app. It'll be put in the chat, so you log into the website and type in the code. You can scan the QR code, and first, you're gonna be voting in word Novo versus yeast. So we'll give you a few minutes here to vote, and yeast is coming out. We can see the drama live. This is good. Basketball. Look at this.
should have muted the screen while people are.
I know, no, it's cool, oh boy, 12 to 11. Oh, no. Half-court shot. Oh. Could be an upset.
Oh, man.
15 to 17, everybody vote, don't forget to vote online. We're waiting a few minutes. 34. Oh, we're doing for a tiebreaker. I think I voted.
Oh, come on, yes. Wow, that's
Come on, someone else, break the tie.
Yeah, I think another device.
What?
Open another device.
Everyone online voted? Coin? Yeah, do you have a coin? Hey Siri, flip a coin, heads or tails? Heads is Novo.
It should be rock, paper, scissors.
It's head, Novo wins. Cinderella story. All right, we're gonna do that again. That was a lot of fun. So in our championship is Novo's Fingobium. For now, we're gonna vote for
Drama, sorry.
against Zymo and Ices. So again, it's a new code, a new QR code, a new.
It should be the same.
No, it's the same thing. Don't listen to me and just vote. Oh, Strep coming out early,
Oh, wow.
pushing the advantage. Whoa! There. Simo struggling, throwing up bricks.
Oh, here we go.
Oh, here we go, coming up, but I think 34 is what we had before, wasn't it?
We had 36.
36, oh, 35? I think that's it, right? All right, so, another upset. I would say I was impartial, but I did not imagine it. It's not a scripted life feed. I mean, I know. So now we're going to have a final round. So now it's the Novo Knights versus the Strep Stingers, I think was your team name.
Yep.
And so we wanted to have just one more few minutes. Any last question? This is a big deal, people. There's a crowd here. So if anybody has any last questions to separate these two very unique microorganisms, both bacteria, then any last minute questions, any final? Daniel?
So like, strep, as in strep throat, right?
Streptomyces.
Yeah, okay. Can Novo get effective in this?
No.
Oh, well, alright.
So what are the safety considerations with working with each of your microbes?
You should always wear proper PPE. As per your biosafety protocol.
Well, we can bring in the other two as well. So
Yeah.
Zymo, you mentioned all these CRISPR tools, all these different tools of putting stuff in. I imagine antibiotic resistance might be involved. Like, what are the safety measures that you have to take to make sure that Zymo doesn't start eating trees or whatever?
Yeah, Zymo is like naturally occurring in our environment without issue, so, not unlike E. coli, we coexist with it happily, and people are drinking it happily, so. It's not getting people sick.
Okay. Yeast? Wow.
Yeah.
You
We
mentioned you
choose
wanted
to drink it.
everything, right?
I mean, you do your classics, you wash your hands, make sure, especially antibiotic resistant genes resistance, we'll try to get rid of that as soon as we can, but.
Novo?
Yeah, so after it's done making all our bioproducts, we make sure to bleach all of the cell material. And we also autoclave anything that has been in contact with our strains as well. Novo itself is not pathogenic to humans or anything like that, so we don't really have to worry about that too much. But again, we are manipulating these strains, and so, we do have to be careful and make sure that everything is disposed of properly.
Someone who has killed his fair share of bacteria in his life, pretty easy. You throw some things in there,
Yep.
you throw it in an autoclave, but it is well regulated, so.
Yes.
And of course, because we're all making bugs to make ethanol or that are ethanol resistant, we can't just use ethanol to kill them,
Yeah.
which is a method that some use. That's why we bleach and autoclave.
Exactly.
Yeah.
So, Bailey wrote us a nice poem, and so for our new
Oh my
finalists, you don't have to write a poem, but like, you think of a couplet or a slogan or something kind of artistic to give us one last impression.
If we both say no.
Am I gonna have to think of mine?
A haiku, yeah.
Oh yeah, haiku, that's a good one.
That's 3 lines.
That's
That's
three, yeah.
But it doesn't have to rhyme. Yes, great.
Yeah, if you both say no, that's fine.
Sing a song.
Yeah. What would be like a song title?
You can say that heat kills them all, right? But which one of these has the highest tolerance That's like, no, count.
For heat?
Yeah.
Yeah, so who can who can do hot yoga tonight?
Do you mean like grow in it or
Survive.
just survive? So with spores, you have to heat them up to like 55, 65 degrees to get them to start growing again, so. But then you normally grow them at cooler temperatures, 30.
So spores, Saccharomyces spores can survive in 55, 60.
Yeah, streptomyces
Celsius.
spores can survive survive the heat.
That's probably gonna be.
That's
right?
Probably,
Yeah.
yeah, Novo does
What's
it
the ideal temperature for each of your?
Yeah,
groups?
ideal is 30.
30 as well.
30.
Boring.
Yeah,
See, if one song could represent your bacteria. what
Oh,
song?
we already know. I might go with Born to Be Wild, but.
there you go.
Yeah, that's a good one.
Forgotten also.
I know,
Yeah,
my
it's
screen is blank right now, like.
We're past the point where we normally take tests, so this is hard.
Like the high, yes, like the highway.
Yeah.
Coming in, I love it.
Just feel bad about the trash.
Yeah,
We have a couple of questions.
Oh,
online.
great.
Yeah. So one question is, which organism has the most variability? And then sort of when converting waste to biofuel, the feedstocks may be heterogeneous and of varied quality. Who can be the most productive without dying or requiring special conditions?
Well, I will say that Novo can eat a lot of different types of compounds at the same time. So for variability, it's pretty good with that. But we are testing different conditions and how well it does under certain conditions too.
How about strep?
Yeah, I mean, it similarly eats a lot of different things. There's a class of compounds that is made when plants break down that are called lignin-derived inhibitors that a lot of microbes find inhibitory. But for some streptomyces, that's actually a signal that plants are being degraded and it kicks up their metabolism because they're like, yay, which is special of them.
Another question online, which individual microbe is the biggest? And then follow-up was, which can make the best free throw?
Novo's free throw is perfect.
Well, biggest I think would be yeast, right?
Yeah.
'Cause it's the eukaryote, so it's 10 times at least bigger than bacteria. But among the bacteria, you guys know how big? Strep,
Strep
right?
grows like in long
Yeah,
filaments
Yeah.
as well, so it's hard to just pull out an individual. I mean, you can, but it's just. So.
basically yeast would just roll over
All of
the
us.
3 bacteria and just forget a free throw, it's just gonna slam dunk.
Yeah.
Maybe use one of his butts.
Yeah, I was gonna say, it's already got the little gun.
Yeah.
One can hold, one can.
Yeah.
Anything else, so we can get to the final voting and
Right, let's see.
so. Now, okay, I think
Oh,
it's
wow.
the same one. Now it's the champion, Novo versus Streptomyces. Go ahead and vote. This is for all the, well, this is for the crown.
Might need to start that.
All the crown.
Here we go.
Mine says.
It should be the
Yeah, ours
same.
is Zymo versus
Oh,
Strep,
really?
not Novo versus Strep.
I'm on semi.
Oh.
Okay,
Yeah, it does.
Simon is just all over.
you might need
Should
to
I go into the menu?
maybe click out of here and go into.
It's like that Super Bowl where the lights went out.
Yep.
Technical difficulties here. This is just building tension, right? This is good
Oh, it's working, yeah.
performance.
Oh, it's
There
working?
we go.
Yeah, you can click an answer and then do skip
Should I do the
question.
present?
Sure,
But for this one, right?
yeah.
Whoa, tied up. Whoa, whoa, Strep pulls ahead. Novo's not giving up. Oh, it's close. Oh, down to the last minutes.
Oh, no.
Strap is up by 3. Someone likes it. I think we had some people leave maybe.
Some people didn't.
Oh, there's one for Novo. Someone else liked it. Let's go. Any other votes?
There's 32, 33, oh, no.
Oh.
34. Might have gone. All right, I think that's it. Congratulations. Have to put the crown on.
Of course I have to put the crown on. If I can.
Like, so maybe five.
But you are all winners, so you all get first place ribbons.
Oh,
These are
Mike, thank you.
amazing. I'm gonna frame.
I know. I promised the deconstruction lab that I would give them a shout out for all the different residues that they make for me to test my streptomyces. So thanks, deconstruction lab.
That was part of your sponsorship deal, you had to say that. Right. So I think that's it for the Ford Energy Forum. I want to thank all of you. I want to thank the panelists. It was fantastic. I had a great time. I hope you guys had a great time and learned a bunch of stuff. So let's thank them one more time. Thank you.